Today's topic is organic molecules. And we're going to talk about organic molecules most of this semester because that is what living things are composed of. There's more to these definitions than this, but this is all we really need to know as far as biology is really concerned. Molecules can be put into either the group of organic or inorganic. Basically, do you have carbon or not have carbon? Now, like I say, it is more to that. If you take a chemistry class, we'll go into more detailed demonstrations because we do have an exception. This is one important molecule that is inorganic, but it does contain carbon. So for our purposes of our class, we just need to know carbon, no carbon, except for carbon dioxide. And so therefore the whole rest of this chapter is going to be about these different biological molecules that are all organic. Carbohydrates, which we talked about in lab on Thursday. So if you remember, here is our glucose molecule. And as we can see, those black marbles, they are what we call carbon. And so this is a biological molecule. It's an organic biological molecule. Or we talked about lipids, and we made some fatty acids. If you remember, that was a chain of carbons and hydrogens. As a matter of fact, this one would be unsaturated because you can see we have a double bond in this one. But the backbone of this molecule is the carbon. So the four molecules we're going to talk about in section 3.2 are going to be carbohydrates, lipids, proteins, and nucleic acids. We built some of these on Thursday. We're going to build some of these this coming Thursday. So biological molecules, some of the ones that we've gone over already. Remember we talked about glucose and fructose. Remember these were both C6H12O6. And whenever you have the same molecular formula but are different molecules, this is that term called an isomer. So glucose and fructose are isomers. Some other examples I gave you were ribose and deoxyribose. And I think I gave you glycerol. These are all examples of those organic molecules. Inorganic, by and large, means we're usually talking about things that form ionic bonds, but not necessarily. Water is a good example of an inorganic molecule. No carbon in here. Or salt, NaCl. Remember that Cl, that C doesn't stand for carbon, that stands for chlorine. That's another example of an inorganic molecule. And also when you're talking about organic versus inorganic, make sure you remember the definition relies on context. And they talk about this a little bit in the book. You've heard of organic food. Organic food means that we process food by a certain way. It's not about whether or not it has carbon or not. So, the key molecule here, as we just mentioned, is carbon present or not? And carbon is the base of all of our biological molecules. So, carbon has an atomic number of 6. So if you look it up on the periodic table, it has an atomic mass of 12, so therefore it also has 6 neutrons, because 6 plus 6 gives us our 12, the mass of the molecule. If it has 6 positives, then it's also going to have 6 negative charges. So if we draw our carbon molecule, we'll have an electron, 2 electrons in that first shell, and then you will have the remaining 4 in the outer shell. So that means carbon is looking to make 4 relationships. Remember, for the second shell to be complete, we need 8. So there are 4 valence electrons in carbon. So therefore it needs 4 to be complete. And we think this is why carbon is the base of it. Because it's looking for so many possibilities, this gives us lots of combinations. Remember, oxygen's only looking for 2, so you can't make as many different combinations with that. And because of this, carbon can form lots of different bonds, and we saw this in our lab. So we saw that carbon could form single bonds. So here we have a carbon atom and a hydrogen, and we can see a single bond between them. Or as we just saw in our fatty acid, you can see that it can form double bonds. It can also form at times triple bonds, although that's not as common. So let's take a look now at the template of how all these biological molecules are going to look. So every biological molecule, molecules are going to be some sort of carbon skeleton, or you might want to call it a carbon backbone. And we're going to put stuff onto that. And those things that we put onto that are called functional groups. So we saw that in our glucose molecule. Here's our backbone. Here's that ring of carbon that we had. And then we stuck things on it. These would all be considered functional groups. Everything that we attach to a carbon molecule is a functional group. Or, as I like to put it, you can think of carbon, this is the Christmas tree. Probably should have done this in green, but here's our Christmas tree. It's the base of many people's decorations come Christmas time. And then the functional groups are the ornaments. The things that we can hang off of it. So we can hang ornaments off of our tree, just like we can hang functional groups off of our carbon skeleton. So, what are some different functional groups? So our functional groups can be seen in table or figure 3.3. So I'm going to need some room here, so I'm going to have to erase this off. So we're just now going to talk about those functional groups. So, functional groups. And this is figure 3.3. These are very important to note, not just for this semester, but you're going to need these in your anatomy and physiology classes. You're going to need this in your micro classes. You're going to need this when you take genetics. So the first one that they talk about there is called the hydroxyl group. A hydroxyl group is simply one hydrogen and one oxygen. So this is the functional group, a hydroxyl group. We just have an oxygen attached to a hydrogen. And if you remember, you put several of these into your glucose molecule. For instance, here's a carbon, and here's an oxygen and a hydrogen attached to it. Here's another hydroxyl group. Here's the carbon, here's the oxygen, and the hydrogen. This here is the hydroxyl group. You're going to find that in what they say here, alcohols. And I'm going to use the word monosaccharides. Need a little bit more room. The book calls this sugars, but sugars means something different in science. So I would like you to get into the habit of when you're talking about small carbohydrates, call them monosaccharides. So we've already talked about our glucose as an example. If you have any kind of alcoholic beverage, there's a molecule that are called ethanol, and it will have a lot of these hydroxyl groups on there. Notice we also have a hydroxide ion, so if we were to add an electron to that, notice hydroxyl and hydroxide are two different things. So hydroxyl is OH, hydroxide is at OH minus. All right, the next group they have on here is called a carboxyl. So you notice that this is abbreviated COOH. One carbon, two oxygens, and a hydrogen. When we get to proteins, the building block of proteins are called amino acids. So in our amino acids, we're going to find carboxyl groups. So we have a single carbon, 2 oxygens, and here's that hydrogen. In a sense, we took a carbon and an oxygen, and we put on a hydroxyl group to form a carboxyl group. So that's a carboxyl. We have an amino group, and this is where amino acids get their name. An amino group is nitrogen and hydrogen. This will also be found in amino acids. This side will actually, ironically, act like an acid. This side will end up acting like a base. Part of the thing about functional groups is they tell you what may happen with a particular molecule. And an amino acid is going to have one side that's going to act like an acid. It's going to pop that hydrogen off, contribute. Amino acid side, the amino side, will actually draw hydrogens towards them to create ammoniums. So that's a quick review of our acids and bases. And to give you an example of an amino group, blue is nitrogen, as we talked about in class. I mentioned it briefly. And then we have 2 hydrogens coming off of it. Next in our list is sulfhydryl. I always want to put a pH in there, but English spelling is with an F, so I'm checking myself. Sulfhydryl. So you can also spell this with a pH if British people will often do that. So you might see sulfur spelled with an F, you might see sulfur spelled with a PH. Both are correct. It just depends where you come from. I tend to, unfortunately, do a lot of English spelling, so I might spell behavior or color with a U, but doesn't change what it is. A sulfhydryl group is sulfur and hydrogen. So remember, here we are, we're talking about one of those highly electronegative elements. This is why I told you to know its highly electronegative elements: nitrogen, oxygen, sulfur, and phosphate. You can see all of these so far contain these highly electronegative elements. A sulfhydryl group is going to be 1 sulfur, in this case the yellow, attached to a hydrogen. We are also going to find this in some of our proteins. So some amino acids. And because it's highly electronegative, this is going to actually help a protein achieve its final state. But we'll come back to that in the protein section. Next is the phosphate group. And this is a phosphate in 4 oxygens. So in this case, this would be a typical phosphate group. So you can see here is our central phosphorus in purple, then 1, 2, 3, 4 oxygens surrounding it. And wherever oxygen has a lone hole, it will, or a valence shell that has an electron in it, will fill in some hydrogen, but don't worry about that. Know a phosphate group as the 1 phosphate and 1, 2, 3, 4 oxygens. And this is going to be a very important molecule for energy and your genetics. Your DNA and your RNA all have phosphate groups on them. Now, one that is not in your book that I also want you to know is called a methyl group. So a methyl group is CH3. And this is in a lot of fats and fatty acids. At the end of each of our fatty acids, we had 1 carbon with 3 hydrogens in it. And that is what will compose our methyl group. So here we have our single carbon with 1, 2, 3 hydrogens. These are what are at the end of every fatty acid. So these are your 1, 2, 3, 4, 5, 6 functional groups. These are the different things that you can put onto your base. This is why we can make different molecules, because we can put different ornaments on. I can pop off a hydroxyl group, and I can then pop in a sulfhydryl group. Or I could pop off a hydrogen, and I can add this functional group. This is why chemistry becomes a very interchangeable system. because we can keep the same base and we can just keep popping different things off and popping them other things on. And this is what is going to happen in a chemical reaction. We're going to take something off of one molecule and we're going to put on something else. Next reaction, we'll take this one off and it will now put on something else. Carbon can accept many different things. So you can see everywhere we can pop something off, we can put something new on, and therefore we get all these very nice, interesting molecules. The other thing you want to take away from this is notice that all of these functional groups are polar. They are all small. They will all dissolve into water, so they're all hydrophilic, and they all contain highly electronegative elements: oxygen, nitrogen, sulfur, phosphate. The only one that isn't polar in this group is this methyl group. So you notice it does not have any highly electronegative elements, so therefore it's going to be non-polar. This is not all of the functional groups that are out there, but these are the 6 that are very important to all the chemistry we're going to discuss from here on out in A and P, and in micro, in every biology class you have. These and how they react and how they integrate with carbon is going to give them their particular and unique chemistry.